Welcome to the show, Dr. Jeff Tomkins. How you doing today?
I'm doing great. Hey, let me fill in a few gaps there that got missed somehow.
Gaps in the fossil record.
Yeah, when I finished my PhD, I actually got hired as a faculty member at Clemson University. They kind of counter-offered. I was actually offered a job at another university, and so stayed there. So I actually worked as a professor at Clemson University for close to 10 years at different levels. So, and I actually also ran a genome center, one of the main genome centers working on various agricultural genome projects in the world. And so I ran that for about 5 years. So,
what compelled you to leave and join Creation Research in 2009?
Well, I had contacts at ICR that were encouraging me to get into creation science. And I was involved in a project called the Gene Project at ICR. And there's actually people from around the country at other institutions involved in this. And we would fly into ICR and have meetings. We did that for a couple of years, and that's sort of what got me connected to ICR and the creation science community. And I was at a good point to leave in my research at Clemson. My grants had wound up. I think I had one grant that had a year left in it, and I had just graduated a PhD student as well. So it was just a good time to make a break because I was realizing that my talents were not being effectively used at a big secular university. They were, basically suppressed. You can't talk about what you really see in intelligent design, or you'll basically get fired. So,
and Clemson's a pretty conservative public university, isn't it, by comparison? No?
Probably in comparison to some of the more northern universities. There were a lot of Christians at Clemson that worked there. In fact, one of my professors when I got my PhD was a Christian, and he taught quantitative genetics, and I took his class, and he retired shortly after that, but he was a very strong Christian and creationist. And there were others there as well that were creationists and intelligent design folks. So, but they pretty much had to lay low while they were working there, and a lot of them began to speak out after retirement or after they left.
Or they might get expelled, as the movie aptly states. Well, Dr. Tomkins, I have an atheist friend from ASU. He's since graduated, got his bachelor's, and I think working on a master's in physics. And he used to love to wear this shirt. His name was Rocco. What's up, Rocco? It said 98% chimp. And he loved to parade that shirt around. It's probably his number one shirt. Besides, he had this other shirt that said, "Yes, I actually am a rocket scientist." But I want you to explain to our listeners, why would an atheist wear a shirt that says 98% chimp? What's the argument behind the shirt?
Well, the argument is that there are regions of the human genome that are between 96 and 98% similar to chimpanzee.
Tell me what that means.
Well, that means those are just selected regions. There's also regions of the genome that are missing in chimpanzee, present in humans and missing in chimpanzee. And then there's also areas where the similarity is much less than 96%.
Let's go super basic. Can you define genome?
Well, the genome is basically the sum total of the chromosomes in your cell in the nucleus. So it basically is the blueprint for what makes you who you are, what causes you to develop into a human in the womb, and what basically runs your cell, or is the basic genetic information to run your cell as an adult. So,
all right, so the blueprint analogy, I think, is helpful for people to get an idea what the genome is. Then when we talk about that, why would it be advantageous for someone that holds to some kind of evolutionary dogmatism to say, hey, man, I'm 98% chimp? What's the advantage in the argument in their mind?
Well, evolutionists need about a 98% to 100% similarity in order for their whole paradigm to work, for a chimp to allegedly morph into a human over 3 to 6 million years of evolution. But now recently there was an article where they reneged on that. Now they say it's more like twice that, 13 million years, but it really doesn't matter. but you need about 98% similarity for that to even work as far as mutation rates and that sort of thing.
So they would acknowledge if you didn't have that, then over any period of time, it just wouldn't happen? Is that what you're saying? The evolution.
Well, they need something that's almost identical for the whole thing to actually look feasible.
I want to try to be as clear as we can. Would an evolutionist actually say that chimps morphed into humans, or would they want to nuance it more? I just want to try to make sure we represent the way they would say it. How would they say it, Dr. Tomkins, as far as the relationship goes?
Well, they would say that we shared a common ancestor.
I think that's important for people to realize. So it's kind of more like we're cousins, ancient, distant cousins in the evolutionary view.
Well, they believe something akin to a chimp, but very much like a chimp.
So an ape-like.
Did actually evolve into a human over progressive stages.
So something that supposedly had both characteristics.
But all of those creatures have since disappeared, so now we're left with chimps and humans.
Okay. So do we concede 98%, or what do we do with it?
Well, overall, the genomes are not that similar. There are regions that are 98%. But think of it like this. When you write computer software, what you do if you're a contractor and someone says, hey, I want you to write a new program for me, and it's a little bit different from all the other programs you wrote, what are you going to do? Well, you're going to use the same code from the other programs you wrote, plus make some new code, plus change everything around just a little bit as well. You don't start from scratch. So it's really easy to explain this to computer programmers. And, of course, as a physicist, your friend ought to know some computer programming and understand this concept. So this is what's going on in the genome, because the genome is genetic code. And we do expect to find things similar between humans and chimps, or humans and mice, or humans and rabbits, whatever. You're going to find some code similarities because the code's doing the same thing. Because we breathe the same atmosphere, we need a lot of the same nutrients, we have a lot of the same metabolic processes. But when it comes to development, what causes a human to develop or a chimp to develop, there's enough differences there to explain that. And overall, the human genome is not 98% similar, is not 98% similar to chimpanzee. What I've found using the best algorithms that we have is that it's only about 70% similar overall. Well, and actually other studies have shown this as well. In fact, when they first started sequencing the chimp genome, they generated over 3 million bases of chimpanzee sequence and published a paper in 2002 and said that they could only get about 2/3 of the chimpanzee sequence to align or to match up onto human. Well, 2/3 is pretty close to 70%. And then and that was using the same algorithm that I used in a recent study that I published in 2013. And another guy actually developed his own algorithm and came up with roughly about the same figure as well. So what I've discovered is not really new or independent. It just matches up with what other people are discovering.
How are these comparisons done? Well, seems like that would have some relevance to the answer.
Well, basically, you have a database that you query a sequence that you're interested in, seeing how similar that sequence is to anything in the database. So, what I did in one study is that I chopped up chimpanzee chromosomes into little pieces and then aligned those or matched those onto a database, which was their counterpart in the human genome. So I would chop up, say, chromosome 1 into little pieces and then match that up onto human chromosome 1.
Okay.
Which makes perfect sense because the chimpanzee genome, this is another little trick that people don't widely hear, but the chimpanzee genome does not stand on its own merits. It's aligned or assembled based on the human genome.
Hold on, that sounds like something we'll need you to explain when we come back here in a second. Talking about human and chimp DNA with Dr. Jeffrey Tomkins of ICR.
I think Vocab asked you to clarify your comment about similarities between apes or chimpanzees and the human genome. Would you just pick up there and give us a very simple explanation?
Yeah, overall, these similarities between the human and chimpanzee genomes are only about 70%, based on the algorithms that we have to work with at this point. And other studies have confirmed this, and my own research has confirmed this. But there are regions that are highly similar between humans and chimpanzees. And then there's regions that are present in humans and missing in chimpanzees and vice versa.
So let me ask a very basic question, maybe even naive. If as the vocab started with the T-shirt, I'm 98% chimpanzee. If that were true, which you're debating, you're saying it's not, then their concept of evolution, whether it's over 6 million or the longer 13 million period, potentially had a chance to happen. If it's closer to 70%, 2/3, 70%, as you've just postulated, then are you saying, I'm asking a question in the form of a statement, are you saying that evolution then is impossible? It could never have happened because they weren't similar enough?
Well, absolutely. Actually, anything probably less than 98%, it makes it impossible.
And both sides agree with that. That's why this is such an important issue?
Right.
Now, of course, even if it was 98%, that doesn't prove evolution.
No, it really doesn't, because another interesting point to make is that when our genomes do vary, they do vary between humans, and they've been studying this all over the planet by sequencing human genomes. They found that this variation is actually highly controlled. It's only allowed in certain parts of the genome where it doesn't really mess things up. And where it has occurred in regions that it can mess things up, they found that, in fact, it has, that these mutations are associated with human disease that's heritable, and there's just literally hundreds of conditions related to mutation. So mutation in and of itself is a whole other topic. And so really, yeah, even if the human genome was 98% similar to chimpanzee, that probably wouldn't even work either, considering the fact that variation is highly controlled in the genome. It's allowed in some areas to create the variation that we see among humans. It's disallowed in other areas. And when it does happen in areas that are off limits, it's usually related to something bad.
I have an article you wrote, How Similar Are Human and Ape Genes? And in there you say that when they do these comparisons, they disregard the non-similar DNA regions. And here's a quote: "Many evolutionary studies have involved the selective use of protein coding segments in the genome called genes." So does that mean this, that a shirt should say 98% chimp according to certain regions or something like that?
Yeah, absolutely. According to certain cherry-picked regions, right? And
so, because 70% is quite a divergence from 98%. So
that's when you take the whole genome into consideration. You don't just cherry-pick what you want. And see, this is another point to use the kind of algorithms where you model mutation, you model evolution. You can't really use anything that's much outside a 98% range. The programs just don't work outside those ranges. You have to get rid of all what are called insertions and deletions, the big ones, and you just can't use those areas. So there is, some pragmatic aspect to it as well. You can't do your evolutionary studies on those areas that don't cooperate.
All right. So with that, you mentioned something briefly earlier I want to ask you about. It has to do with the amount of time needed for the changes. So let's just say that, you only kind of had 2% to go or something like that on an evolutionary framework even. I have a book here put out by the Discovery Institute called Science and Human Origins. In the first chapter, written by Ann Gauger, she has a subtitle in it that's this: "So much to do, so little time." And it talks about the number of mutations needed And even in the evolutionary time frame, do you actually have enough time? Because for every sort of anatomical change you would see on the outside, there's dozens of things happening sort of beneath the surface. I mean, I'm explaining it roughly, but help us understand, even granting you've only got 2 to 3% to go, you've got all these years, are there still problems?
Well, the problem is that when we talk about genes don't operate by themselves. Genes operate in networks of other genes and subnetworks and networks of subnetworks. And the genome is highly complex in how it operates. And so when you affect one gene, if you mutate it, then you affect the performance of every other gene connected to it that produces a protein that interacts in that network. And so when you affect one little part of the system, there's implications across the other aspects and networks of that system that are connected to that.
So there's a domino effect, as it were.
Yes. So how do you coordinate these mutations across all these networks? In fact, that's the big area of study right now in biomedical genetics.
Okay.
Is network biology and how networks operate in the cell and, of course, in the genome.
And so we all know that, evolution doesn't have foresight. And so if it's...
Well, it's really hard to think about evolution in light of network biology. It's very difficult to model it and think about how it actually really might work. Now, there have been some papers that have tried to do that, but it's highly theoretical and a lot of theoretical mathematics.
All right. Well, when we come back, we for our next segment, where we're winding this one down, I'd like to ask you if this is the case, what do you see as the future in this area of research on the evolutionary biologist side and the biochemist guys and the geneticists and all of that? I'm curious what you think will happen, because it's not like they're going to give up or something. And then also, I'm curious, then why is this? When I talk to an evolutionist, the first thing they say to me, the informed guys, is that you need to check the DNA evidence. That is the strongest evidence. I want to ask you why they think that is, Dr. Tomkins. Talking to Dr. Jeffrey Tomkins of ICR, Institute for Creation Research. And we've been asking questions about this claim evolutionists often make, 98% chimp. And the idea is that there's so much similarity between the human genome and chimp genome that it's leftover evidence that really we are all related, evidence for evolution. And Dr. Tomkins been showing us that, first of all, the claim is overblown. And second of all, even if you had that much similarity, you still would have a lot of work to do. Do you want to summarize anything else before we get into the next question of why then is still this one of the key arguments that I hear, at least on a personal level, to the point where it's popular on T-shirts? And what do you see the future for this kind of research? I'm curious.
Can I ask a question to direct that?
Yeah.
Okay. So, Dr. Tomkins, if, for example, you won the case and you and your colleagues and proved that only 70%, 2/3, 75 in that range were similar, not the 98, would would everybody fall down and admit creation and deny evolution, or what would they do if they lost that argument scientifically? How would they respond?
Well, they would probably propose some mechanism that would cause these big deletions, these big insertions, these major structural changes in the genome. Because if someone wants an excuse not to believe in God, they're going to come up with anything. But I think that they're going to have to come up with some sort of an excuse at some point, because the DNA sequencing technology now is rapidly improving to the point where they can sequence much larger chunks and then assemble those back into chromosomes. So right now, the kind of technology that's being widely used only produces DNA fragments that are about 500 to 600 bases long. And then those those little fragments have to be reassembled into something that makes sense. And now the sequencing technology is getting to the point where they can sequence 10,000 to 30,000 bases and then assemble those fragments. And
so you're saying
it's much more accurate.
It's gonna happen where they're going to lose this argument. Is that the gist of what you're saying?
Well, there's a company called PacBio that is now producing these sequencers, but they're so expensive that very few places can afford them. Only your big genome centers can. But when this technology really begins to enter the marketplace, I'm hoping that they will start resequencing the chimpanzee genome and coming up with much more accurate, what we call in our business, assemblies.
Vocab, that was a very interesting answer Dr. Tomkins gave. When you don't want to believe in God, you're going to come up with some reason not to. Different subject, but somewhat related. Back in the early days after the Roe versus Wade Supreme Court decision allowing abortion, 1973, those early years, the pro-life community thought all we need to do is prove that this really is a human life. And technology, even the ultrasound, wasn't nearly as developed. By and large, that argument has been completely proved. I mean, the fetus is a human. It's not a chimpanzee or an elephant. But and we thought at that time, we win that argument and we're going to crush the opponents. Well, they just changed, we won the argument, but it didn't make any difference. They just came up with different reasons to argue for abortion at any point. And so you got to win the argument. Truth always should win out. But I thought Dr. Tomkins' point was really right. Once we win this argument, they'll just come up with a different way of approaching it.
Yeah, well, that's where presuppositional apologetics and methodology come into play. But I mean, I want to be precise with this argument. It's really this show is more of a polemical show in the sense that we're attacking a specific argument. Everything fits together, but this specific show itself, we're not necessarily saying, here's why Christianity, here's why creation. Now, again, I would talk about presuppositionalism and the whole realm of evidence and all that kind of stuff, but that's a different discussion. This particular thing is saying, are these claims true, and what do they even get you if they were true? And we're going to talk a little bit more about that when we come back with Dr. Tomkins. Human chimp DNA, what would you consider some positive evidence for the fact of creation versus just an answer to evolutionary claims? Maybe we share some of those with the listeners, Dr. Tomkins.
I think one of the really exciting areas that's coming out in genome sequencing, and it's an area that I worked in as well before I left the university, and that is that every time an unusual creature has its genome sequenced, we're finding unique sets of genes in these creatures that are specific to that creature. It's like just a blueprint specific to that creature. They don't know where these genes come from. They have no evolutionary history to them. Some people call them orphan genes. Other people call them taxonomically restricted genes, meaning they're just restricted to that type of creature. And I noticed this when I was running a genome center, when we would extract the RNA from a creature. So when DNA, the genes in the DNA are copied into RNA, and then the RNA is turned into proteins, or a portion of that RNA is. And so you can actually pull the RNA out of a creature's cells and convert that into DNA and sequence that, and sequence the genes directly. And so we would do that for all sorts of strange things, plants and insects and shrimp and oysters, and we did all kinds of unusual creatures and plants. And we would find that when we would take those gene sequences and compare those to the public databases to try and find out what in the world we were sequencing, many times, well, actually most of the time, we would only get about half of the sequences would match up to anything in the databases. And we were always kind of wondering what was going on, but obviously these were unique blueprints specific to these creatures. And since I left the big university in 2009, I'm seeing more of these papers being published where they're sequencing unusual creatures' genomes and discovering all of these orphan genes that have no evolutionary history. They just pop up suddenly in these creatures. They don't know where they came from or what they evolved from. They're unique and specific to whatever creature that they're studying.
And so we use that as an argument for creation. The evolutionists do what with that data?
They don't know what to do with that data. So some of them try and attempt to invoke really wild mechanisms like horizontal gene transfer. So somehow these multicellular creatures extracted genes from each other through some sort of a mysterious mechanism or vector. Now we know that occurs in bacteria, and we know some of the mechanisms that occurs in bacteria. And we also know that some bacteria can transfer their genes to their multicellular hosts that they live in a biological relationship with. But as far as just multicellular creatures wholesale exchanging genes with each other, it's just not happening.
So they would need to do that because these unique traits and features don't match a common ancestry paradigm. Is that right?
They can't be transmitted through the sexual lineage or through sexual reproduction. They just
got to get it some other way.
They show up, and where did they come from?
Well, what about this? Other people would say, well, you guys aren't talking about everything. Shared pseudogenes are also a strong evidence for common ancestry. What about that? What about these pseudogenes? Because I know you've written on that as well, is why I asked that.
Well, it turns out that the more pseudogenes, so-called pseudogenes, that they study are functional and required for life. In fact, many of the pseudogenes that they thought did nothing before, they're finding out that if that gene is mutated, you get cancer. So pseudogenes don't produce a protein product. They produce what's called a long non-coding RNA.
So non-coding regions of DNA, some people have nicknamed them, although this name has fallen by the wayside recently because it's ultimately inaccurate, junk DNA. That's the more technical name is pseudogenes. You're saying we're finding out they do more and more, we discover, have a function. But what about the fact that if they're shared between two species, what if someone says, well, there's more evidence?
It's more evidence of function.
Okay. What do you mean by that? Like, what's the answer to someone saying pseudogenes being shared is evidence for common ancestry?
Well, even evolutionists will say that if two organisms have the same gene or pseudogene, that there's a strong possibility of function. Because something that's conserved or similar between organisms means that mutation hasn't had time to play havoc in it, that it's constrained, it's being selected against, it's being selected for, and so it's not changing. It's not neutrally evolving. It's functional. So, but pseudogenes are very important parts of the genome. We're finding out that as we study them one by one, that they produce RNAs that actually interact with the protein coding counterpart of that gene. So this gets a little technical, but a lot of they're called pseudogenes because they have some similarity to another gene in the genome that produces a protein product, but the pseudogene does not produce a protein product. But basically, the RNA that is copied from a pseudogene acts in sort of a competitive binding network with little RNAs called microRNAs that helps control the transcripts or the RNAs produced from the protein coding gene. So they operate in these complex networks with each other. So that's why when you mess up a pseudogene, it typically can cause a disease or cancer, and it's involved also in the same kind of network that the protein coding gene is involved in.
Right, so you don't want to monkey with those. Haha.
No pun intended.
Right. Dr. Tomkins, a lot of the work I've seen you done, some of it's written at sort of an educated lay level reader, it looks like. And a lot of what I've seen involves sort of a meta critique or a meta analysis, cooling lots of different data together. aggregating the data and comparing it, and then offering critiques of the evolutionary paradigm found therein, sometimes even from what you might consider evolutionary sources themselves, where they say, hey, guys, we need to be more precise. Is that an accurate description of a lot of what you've done with ICR thus far, offering that sort of critique aspect to the data that's out there and giving a different interpretation on it? Help us understand kind of that role that you do and how that kind of research plays a role in the larger scientific schema.
Right. Well, when I see research that's published, I'm interested in the data. What kind of data is being discovered, and what does that mean? And, I'm open to whatever it means. But if it's not a hypothetical type study, a lot of evolutionary studies are just totally hypothetical. There's really no empirical science going on there whatsoever. It's totally theoretical. But when I see papers published, even though it might be some sort of a biomedical paper, or they're discovering some new feature or function in the genome, I look at it from a design perspective, because it's obvious what's going on here. Everything's designed. Everything's incredibly complicated, and it gets more complicated every single year that we delve into the mysteries of the genome. And so I do have that perspective where I believe that there's a omnipotent creator that created everything, and it slaps me in the face every time I read a research paper. I mean, it's really undeniable. And I believe that there's a lot of scientists out there that really are not speaking out. They know what's going on, especially these guys in the biomedical area, in the ENCODE area, which is a very diverse area. And these guys know exactly what's going on. They just can't say anything because they want to bring home a paycheck every couple of weeks. So,
well, Dr. Tomkins, can you tell people, if they want to track down some of your articles online and maybe some of your published works, where to find them, how to get a hold of them, maybe some of the titles you would recommend? Where can people find out more about you and your work?
Well, I try and keep up with, the top-level genomics, genetics, ENCODE-related research on the ICR website at icr.org. And so I like to write up these little news articles at a semi-technical level. And so you can basically go to icr.org, type in my name, Tomkins. And so I try and write these little news articles. They're about 600 words, so it's kind of a small read. You can do it, fairly quickly and keep up to speed that way. And then I also have technical things that I publish in various journals. The Journal of Creation is one. You can go to their website, just type that in, Journal of Creation, then go to the link and type in my name there. Or the Answers Research Journal. I've published quite a bit of my research there. And, of course, this is all at a technical level, but there may be some people out there listening that would want to see those articles. You can go to the Answers in Genesis, and then there should be a link there to the Answers Research Journal, or just type that into Google and then go directly to the link and then type in my name, and you can pull up my papers there.
All right. Well, thank you very much, Dr. Tomkins. Appreciate you joining us today, Pastor Bob.